Hi, my name is Aleix Lafita and I'm a fourth-year PhD student with Alex Bateman at the European Bioinformatics Institute in Cambridge. My PhD project is focused on the study of globular domains that form tandem repeats in proteins. Within these highly repetitive proteins, we discovered many cases of structural rearrangements, where native contacts at the domain core are conserved, but the topology of the protein chain is not. Some examples are circular permutations, domain swapping, and domain atrophy, which I will be discussing later on. Today I am presenting new work recently published to create a lightweight and flexible modeling approach for these structural rearrangements, which is based on the concept of Euclidean distance matrices, or EDMs. EDMs are matrices of square distances between points in a Euclidean space of arbitrary dimensions. They have many useful properties and applications in different fields, including molecular conformation. The atomic coordinates of a protein are points in 3D, here shown with consecutive numbers. So it is straightforward to compute their distance matrix, here shown as a heat map with dark colors for small distances. Inverting a distance matrix into a set of points in 3 dimensions can be done using a technique known as multidimensional scaling, but it can be challenging, especially if the matrix contains noise or missing entries. Based on these concepts, we propose a modeling approach for structural rearrangements as following. First, the atomic coordinates of a protein, here on HS3 domain, are converted into a distance matrix, shown here. Next, the entries of the distance matrix are rearranged according to the structural changes to be modeled. Here we show a circular permutation at the position of the red arrow, here and here. The red boxes shown here are conserved parts of the matrix, which will be swapped in the transformed distance matrix. As a result of the matrix rearrangement, some entries will be unknown, here shown as white stripes. So a matrix completion algorithm is used to fill the unknown entries of the matrix. Shown here, the final completed rearranged matrix is converted into the 3D points corresponding to atomic coordinates using multidimensional scaling. An additional detail of this modeling approach is the use of atomic distance bounds from experimental structures in the PDB. This is in order to conserve protein geometry in the models. More details are explained in the article. Shown here are 4 structural rearrangement examples where EDM modeling can be applied. Models are represented as rainbow backbone, and experimental structures are superposed in pale cartoon. First, a circular permutation of a YIBK medial transferase requires a linker of 6 residues between the main termini, shown here. This is correctly predicted by the EDM model, as compared to the experimental circular permutation. The next 2 structures, cyanovirin N and EPS8, are domain swap dimers built using their monomeric domain structures. As seen, cyanovirin N appears to be rigid, so there is little flexibility between the orientation of the 2 domains, and the model correctly predicts the orientation of these 2 domains. On the other hand, EPS8 domain appears to be flexible, showing many different domain orientations between the 2, which do not necessarily correspond to the experimental one. Finally, when modeling the domain atrophy in RIPs, the loss of 2 beta strands in RIP long leaves part of the domain core exposed, as it can be seen in the model here, so this part is deleted. This gap is compensated by an alpha helical turn in the experimental structure of the atrophied RIP domain. These EDM models are coarse-grained and solely based in protein geometry. Although they might be too simple for some applications, several programs can find this modeling alternative better suited to the task, as is the case for the examples just shown. Creating EDM models takes only a few lines of code and runs in just a few seconds, making it easy to use and customize for each specific application. Code written in R can be openly accessed on GitHub, and many more details can be found in the article here. Thank you for your interest. If you have any questions, please contact me at the email below or meet me during the virtual poster session. Goodbye.